Oops. Let me get my. Physiology is one of my favorite topics because it covers so many different areas of biology, but it is particularly important for clinical medicine, which I know you are studying. And so to have a good understanding of physiology means that you will become a better doctor. So I don't know if any of you are together where you can talk to each other, but if you are, I will be stopping a couple of times and asking you to think about something or to look at something and see how you would answer it. So my first question for you then is, which organ system is the most important? So take a second. I've listed 10 organ systems here. And I would like you to think about that and come up with your answer. All right, let's see. And my answer is there is no one system that is the most important. You might say the nervous system because if the brain dies, the body dies. But you could say the same for the cardiovascular system. So, or the respiratory system for that matter. So, all of these systems are so interwoven and integrated that it's difficult to say there's one system that is the most important. You've been studying physiology, and we're going to focus today on the integration of systems, but you've been studying the systems in a linear fashion because time is linear. So when you started your module, perhaps you started with cell physiology, and then you looked at the three control systems, nervous, endocrine, and immune. And then perhaps you moved on to the cardiovascular system, the respiratory system, the renal system, and then perhaps ended with the digestive and the reproductive systems. So you have taken them in a linear fashion. But physiology and physiological function is not linear, it is integrated. And so you must learn to think about physiology as you study it in an integrated fashion, because that's how you will experience it when you become physicians. So today I'm going to talk about the integration of physiology. And we will start with core concepts. And this is sort of a new term that has come into being. Another way of thinking about it is the big ideas in physiology that cross systems or repeating patterns that you will see showing up over and over again. Then we will talk about homeostasis, which is the overriding, to me, the overriding core concept for physiology, because homeostasis is the basis for all of the other core concepts. And then we will take a look at two physiological examples where our pathophysiology shows just how integrated the physiology is. Once you have developed a broad understanding of the organ systems, it's much easier for you to see the core concepts and also see the integration between the systems. So I have taken 8 core concepts, and these are patterns that you will see over and over repeated in different systems, perhaps with a slight difference between them, but it's the same pattern over and over. As we said, homeostasis, I think, is the most important. We also have communication, excuse me, communication, compartmentation, dividing things into separate compartments, molecular interaction, gradients and flow, energy, mass balance, and structure function relationships. So we will take a quick look at each of these now, and then we will start looking at some examples. So, structure function is important because physiology spans many different levels of organization and every level that you find within the human body. Everything from atoms and ions to molecules, cells, tissues, organs, the organ systems, and finally the organ systems functioning together. The compartmentation of the body then is part of the levels of organization, where we see, for instance, in cells that we have obvious compartments bound by membranes. So the mitochondria, the nucleus, the Golgi. But if you look functionally at the cell, there are also compartments without membranes. You can see, for instance, with cell signaling, that a calcium signal coming into the cell doesn't immediately go throughout the whole cell. It may be restricted to a small area, which is a compartment without walls. And then we move up the levels of organization to our body fluid compartments. And for medicine, body fluid compartments are one of the most important things for you to know and understand. So we have our intracellular fluid, or ICF, and then we have the extracellular, or outside the cells fluid, which consists of two subcompartments, the interstitial fluid directly in contact with the cells, and then the blood with the blood plasma being a fluid compartment that is responsible for helping communication of substances throughout the body. And then finally, we can consider the body itself to be a compartment where we are one compartment and everything around us, the external environment, is another compartment. And we are an open system. There's a constant exchange of material between this external environment and the internal environment. And that then leads us to our next core concept, which is mass balance. Everything that is in the body at a given time is a result of what has been there, plus what is coming in, minus what is being lost. And to keep homeostasis, or to keep a constant level, mass balance, there must be balance. What comes in must equal what goes out. And when they are out of balance is when we run into problems. Here's another way of looking at the fluid compartments. In this one, we have all of the cells together in the middle, surrounded by the internal environment that is the extracellular fluid. And then you see the white outside is the external environment. So everything that comes into the body goes into the extracellular fluid, and that is the buffer between the cells and the external environment. Here's another way of looking at this diagram. This is a box diagram that shows the different compartments as boxes. So on the left is the external environment, and the two boxes on the right represent the body. And notice that everything that comes into or leaves the body must cross an epithelium. And the epithelia help regulate what enters or leaves the body. And to get across the epithelium, a substance must then go across two cell membranes. So you're actually crossing a cell layer to move from external environment to the extracellular fluid. And then from the extracellular fluid into the cells is simpler because a substance will only cross one cell membrane. So thinking about mass balance and homeostasis, we have to be aware of the external environment, but also what is happening in the extracellular fluid. Here's another way is that, can you hear me?
Yes, we can.
Okay, I'm getting an internet connection unstable and the translation stopped for a minute. Okay. So the important part of this is to understand that even though some of our organs are inside our body, their lumens, the insides of these tubes and organs, is actually external environment. And so you see on the left of the diagram the digestive system, which is really a tube that runs through the center of the body with input through the mouth and output through the anus. And then notice also that the respiratory system, the lumen of the respiratory system, is external environment, which is why we have so many specialized cells, the mucociliary escalator that is meant to trap foreign particles, and many immune cells and signaling cells that are telling the body what is in that air that has come in. And the same thing with the kidneys. You might not think about it this way, but as soon as a substance filters from the plasma into the lumen of the lumen of the nephron is external environment. And as you know, with glucose, when it's filtered and it cannot be reabsorbed, it leaves the body. It is in the extracellular fluid from the moment it filters into Bowman space. Now, the way to think about this is that lumens are like a hole running through a bead. They're inside us, but their lumens are external environment. And now we move to the core concept of gradients and flow. The simplest is the bulk flow of fluids, and that means air in the respiratory system and blood in the circulatory system. And in order to create flow, we must have a gradient. And for the bulk flow of fluids, we have to have a pump. So that's the heart or the lungs, the thoracic cage moving the lungs. And so this is bulk flow of fluid down a pressure gradient. But there are other types of gradients. We have concentration gradients, we have osmotic gradients, and we have combined electrochemical gradients. And so these then also are places where we can see flow. So, for instance, this is where transport across membranes, whether it is across two membranes for an epithelium or simply one membrane to go in and out of a cell, there must be a mechanism for moving the substance across the membrane. And so those transport processes, the simplest, if it is a lipophilic or lipid-loving substance, is simple diffusion, something that can actually move through the phospholipid bilayer. If a substance cannot go by simple diffusion, however, then it must go either by protein-mediated transport, and that means our channels that have an open pore or carriers that actually bind to the substance being moved. And if a substance is too big to go on a transport protein, then we must go by vesicular transport. And that means endocytosis, exocytosis, phagocytosis. And now, one of the most exciting in the research area is extracellular vesicles, parts of cells that are pinching off and being transported through the extracellular fluid from cell to cell. And I think in years to come, we will see and learn how important extracellular vesicles are in communication. But the thing to remember here, we talked with bulk flow about flow down gradients, but flow can go across a gradient, but it requires the input of energy. And so you can think about the sodium potassium ATPase that is moving the ions against their gradient, but using energy from ATP. And so that now brings us to our next core concept, which is energy. And what are the energy sources in the body? When we think about energy, we have to acquire energy, which we do because we are animals. We acquire it through our food. And then we need to extract it and we need to store it. And so there is a lot of potential energy that we store in chemical bonds, in ATP, in other high energy bonds, as well as in biomolecules, such as glycogen and lipids. But there's another source of potential energy that sometimes we don't remember is actually potential energy, and that is the energy that is stored in concentration gradients, electrical gradients, and the electrochemical gradients that we use for electrical signaling, particularly in the nervous system, but not restricted to nerves and muscles. The release of insulin, for instance, begins with an electrical signal that it comes about because the pancreatic beta cell is able to sense the concentration of glucose and uh, use a gated channel to change the electrical signal of the cell. And when we start talking about all of this, now we're getting into one of many forms of molecular interactions. So, protein binding is universal and it occurs over and over. And for many years, we talked with enzymes about the specificity of an enzyme or the saturation of an enzyme or competition between substrates for an enzyme. But those are not limited to enzymes. Cell receptors for signal molecules have the same properties, as do the transport proteins that we talked about, particularly the carriers that bind to their substrate. So, protein binding is a big universal theme that you will see over and over again. And it's important because small changes in a protein can significantly change their binding properties. And a nice example of this is maternal and fetal hemoglobin, which have very different oxygen binding proteins. So the fetal hemoglobin has a much higher affinity for oxygen than the maternal hemoglobin. And this is due to a change in the protein structure. So we have covered all of 8 of the core concepts, and I want to show you a figure. This is a figure that you might have studied in the respiratory system, and it's looking at oxygen transport on hemoglobin. So its total arterial oxygen content is consists of the oxygen that's dissolved in the plasma, as well as the oxygen that binds to hemoglobin. And then hemoglobin is going to be the percent saturation of hemoglobin plus how much hemoglobin is there, the total number of binding sites. And you can see some of the factors that you studied when you looked at the hemoglobin, oxygen hemoglobin saturation curve that will shift or change the saturation. So PCO2, pH, temperature. And then on the other side, how much oxygen dissolves in plasma is a function of 4 primary factors. What is the composition of the inspired air? What is the alveolar ventilation? And you learned how to calculate that if you've done respiratory already. How well can oxygen move between the airspace of the alveoli and the blood? And there are some factors there. And then finally, is there adequate blood flow to the alveoli? So I want you to take a minute. I'm going to be quiet, and I want you to see there are four of the eight core concepts are represented in this figure. So I'm going to be quiet and give you some time to think about it, but you can mark where they are, if you will, and think about where they are. Okay, let's see how many you were able to find. So the first one that I'll show you is gradients. Moving oxygen between the alveolar airspace and between the environment and the alveoli is all the movement down a gradient. Alveolar ventilation is that bulk flow of air that we talked about earlier. And then the oxygen diffusion between alveoli and blood depends upon the gradient, the concentration gradient for oxygen, which is able to move by diffusion. All right, so there's one. Here's two physical properties of the system. So, resistance of the airways, how compliant is the lung? Is it stretchable or has it become stiff with fibrosis? What is the surface area of the alveoli? Has it been lost in a disease like emphysema? And what is the distance for diffusion? Oxygen is not very soluble in water. So if you have something like pulmonary edema, it is going to slow down the diffusion. So those are all physical properties, structure, function. And then on the other side, we have protein binding. I even showed you the curve for that. So I hope you all got that one. So oxygen binding to hemoglobin, and that's a molecular interaction. And then finally, homeostasis, because the body has to maintain homeostasis of several of these parameters. So the PCO2 of the blood, the pH of the blood, and the temperature are all homeostatic factors that can be helped or disrupted with the respiratory system. So there are four different examples of core concepts within a single system. So the one we haven't talked about is communication. And communication is important because without communication, the different systems cannot integrate and work together. So we have our local communication, gap junctions that take things directly from one cell to another cell, such as the gap junctions in the heart that allow electrical signaling to pass directly from myocardial cell to myocardial cell. We have diffusion to neighbors. We have contact dependent signaling where one cell literally binds to the surface of another. And we see this primarily with our immune cells. And then paracrine signals, autocrine signals, and cytokines that are acting locally. And I forgot to add, but I would put extracellular vesicles now there as one means of local communication. And then we also have long distance communication, and that is primarily done with chemicals moving through the blood, both hormones and cytokines, and then the combined electrical and chemical signals in the nervous system. So the nervous system sending long distance electrical signaling that is converted to a chemical signal of a neurotransmitter or a neuromodulator at the synapse. So, two patterns, local and long distance. Cell signaling is very interesting because there are some patterns that repeat over and over. So, one is, where is the receptor? If we have a signal molecule, where is the receptor? And it can be on the cell surface, and this is our idea of signal transduction and second messenger systems, or the signal molecule, if it can enter the cell, becomes lipophilic going in, and then it can bind to a receptor either in the cytosol or even on the nucleus. And in general, the cell surface receptors trigger a much more rapid response, while the intracellular signals tend to go to the nucleus and cause the release much slower synthesis of new proteins. But what we're seeing now is that some of our signal molecules, like insulin, that are traditionally thought of as binding to a cell surface receptor, they do, but they also have the ability to have slower onset, slower onset, longer acting intracellular changes. And some of the steroid hormones that we think of, particularly estrogen, will then be able to have a long-term effect with a nuclear receptor, but they also have cell surface receptors and are able to initiate some rapid responses. Another pattern that we see is that a single signal molecule can have different effects. And this all depends upon what the receptor is and what the intracellular pathway, what the signal transduction pathway is at the target cell. So here's our example. We have epinephrine from the adrenal medulla coming out, and if epinephrine binds to an alpha receptor on a blood vessel, it initiates the same response that norepinephrine does, and you get constriction of the blood vessel. But if epinephrine binds to a beta 2 receptor on a blood vessel, then what you see is vasodilation. So one signal, two opposite effects that are determined by the receptor, not by the signal. Another pattern, and this is one that is sometimes difficult to remember. It's much easier to think about it's on and now it's off. Oh, yes, it's doing this, and if it's either doing it or it's not doing it. But many physiological processes are what we would call tonic. It's like the volume on a radio. You can turn it off, but when it's on, you can regulate it up and down. So you can actually leave it on all the time and then only choose to turn it up and hear it sometimes. So here's our example of tonic control. And this is actually your sympathetic control over blood vessel diameter. So we just saw epinephrine. Now we're looking at norepinephrine. And essentially, the sympathetic innervation of the blood vessels is there all the time with a medium signaling rate. If you decrease the signal rate, you put less norepinephrine on the cell, then the vessel dilates. If you increase the signal rate and put more norepinephrine out, then the blood vessel constricts. So this is an example of tonic control. And then finally, we have antagonistic control. So if two signals have opposing effects, they are said to be antagonistic to each other. They don't have to work through the same mechanism, they just have to have opposite effects. And so here's our example, again from the cardiovascular system, where we know that sympathetic neurons are able to speed up the heart rate, while parasympathetic neurons decrease the heart rate. And they are tonic. The parasympathetic control over the heart rate is tonic because in the absence of innervation, the autorhythmic cells go at about 100 beats a minute, but our typical resting heart rate might be 70. So we know that there is tonic control of heart rate being exerted by the parasympathetic division. Here's another example, insulin and glucagon, where you have glucagon dominating in the fasted state and insulin dominating in the fed state. And they have opposite effects working through very different signal pathways. So the communication then requires control systems. And we have three control systems. For many years, we thought only of the nervous system and the endocrine system. And for a while, they were separate. And then we learned that they overlap a lot. We have neurohormones, we have the nervous system controlling the release of classic hormones from endocrine glands. And then after COVID-19, we realized that the immune system really is one of our third control systems. And so here is a figure that shows that, where the endocrine system is putting out hormones that can act on both immune cells as well as the nervous system. Nervous system can put out neurocrine molecules. That means a neurotransmitter, a neuromodulator, and neurohormone. And those neurocrine molecules can act on the endocrine system and the immune system. And then the immune system is putting out cytokines. And as we saw with cytokine storms during COVID-19, they can have effects on many different systems in the body. So now we come to our second part, which is homeostasis, the final and really the overriding core concept for physiology. And homeostasis is defined as the maintenance of a relatively constant internal environment. And so that means we are trying to maintain the ECF, that's our internal environment, relatively constant. And it's very simple. If you have an organism in homeostasis and something happens that results in loss of homeostasis, there will be an attempt to compensate. And if compensation succeeds, it's good. But if compensation fails, then this is where we run into illness and disease. And this is where you will be working in the years to come. So you need to think about and understand what has happened to homeostasis in each of the conditions you see, because very often the best way to treat the condition is to try and restore homeostasis. So we're back to our figure again, simply showing you that the extracellular fluid is that buffer, and that's where we monitor homeostasis. Very simply, homeostasis of the extracellular compartment, everything about it is aimed at keeping the brain functioning. The brain is our central control system. If the brain goes down, you're dead. So all of the things that are monitored in the extracellular compartment then are aimed at keeping the brain going. And we have 10 key regulated variables. So a regulated variable then is going to be the part where if as long as we're in homeostasis and the variable is within the desired range, and you see the up in the icon there at the top right, you see the dashed line that represent the acceptable range. It's fine. There's variability within it, but as long as you're in homeostasis, you stay between the lines. But if something happens and the variable moves outside the desired range, this will activate a sensor. And that sensor then will send a signal to an integrating center. And the integrating center will then initiate a response that goes back and tries to bring that regulated variable back into range. So there are 10 key regulated variables, and they are in the extracellular fluid. And so I want you to see how many of the 10 key regulated variables for homeostasis you can list. So I want you to, if you have a pencil and paper, please go ahead and write them down and you can then check yourself. Okay, let's see how you did. This is the difficult part of doing this over Zoom because if you were in my class, I would be watching you and I could tell how far along in your answers you were. But we will go ahead and you can now compare your list to my list. So here they are: blood pressure and blood volume, which are related but not identical, oxygen, blood oxygen levels, and blood CO2, plasma glucose, pH, osmolarity, the concentrations of two ions, potassium and calcium. And you notice sodium is not there. And we can think about that a little bit later. And then temperature. So these are all parameters that variables that you will find in the extracellular compartment for which the body has mechanisms to try and keep them in homeostasis. So, what is a regulated variable? And simply stated, a regulated variable is a variable that is monitored by a sensor. In other words, the body has a sensor that is keeping track of the level so that if it goes out of range, the sensor is activated and can start a response. Some sensors are tonically active, always monitoring. We talked about tonic before. And a good example of that would be the carotid and aortic baroreceptors. If you monitor the neurons coming off the receptors, you'll see that there's always some level of activity, some tonic level of activity. A few other receptors, however, are not active until the variable goes out of an acceptable range. And so, for instance, our hypothalamic osmoreceptors that cause the secretion of antidiuretic hormone, vasopressin, if the osmolarity is below a certain level, the sensor is not activated at all. It is only activated when osmolarity goes out of range above what is desirable. So, intermittent activity versus tonic activity. So, What does it take? We can talk about a reflex where we have a sensor, an input signal, integrating center, output signal, target, and response. The knee jerk, the knee jerk reflex or patellar tendon reflex is a reflex, but it is not a homeostatic reflex. So, to be a homeostatic reflex, it must meet two requirements. The stimulus must be a change in a regulated variable, a monitored variable, and the reflex has a negative feedback loop that shuts off the reflex when the variable returns back within the normal limits. So you can see here in the figure that if it goes a little bit above or a little bit below the acceptable range, which is that light green bar, then the response loop turns on. And then once it comes back into range, the loop turns off. And so we say that is a negative feedback. It shuts off the reflex. Not all feedback is negative feedback, though. We said that the response then decreases the stimulus and shuts off the loop. But we also have a few positive feedback loops, and these are not homeostatic because an increase in the response just makes the stimulus worse and it's positive, it's spiraling out of control. And the only way to stop a positive feedback loop is to have some sort of outside factor that happens. So positive feedback is not homeostatic. So, how is a regulated variable different from a controlled variable? And these are two words that we often use as if they are the same thing. But for homeostasis, they're very, very different. And so let's take a look at an example so you can see the difference between a regulated variable and a controlled variable. And our example is this map of the baroreceptor reflex. So you see the yellow circle at the top says increase in blood pressure, and that goes out of range. It is monitoring the pressure and it will send that information to the cardiovascular control centers in the medulla oblongata, and they will make the appropriate response through the autonomic divisions. And you will then have, with the increase in blood pressure, you get vasodilation from the arteriolar smooth muscle, you decrease force of contraction, and you decrease heart rate. These will then decrease peripheral resistance and cardiac output, and blood pressure will come down. And as blood pressure comes down, we have the negative feedback loop that stops the firing of the baroreceptors because they're no longer sensing high blood pressure. So, what is the regulated variable in this example? It is the monitored variable. It is the one that the sensors are monitoring. And so that is the blood pressure. So the change in blood pressure is the regulated variable. What are the controlled variables? Well, it turns out the controlled variables are on the output side of the reflex. They are the parameters that are being changed by the reflex. The variables are the peripheral resistance, in other words, vasodilation, and then cardiac output. And so the controlled variables, the important thing to remember is that controlled variables don't have a sensor. So we would say in the baroreceptor reflex, the body regulates blood pressure by controlling heart rate, force of contraction, and vasodilation. And so you can always tell a regulated variable because it will have a sensor. Our body has no way to know what the heart rate is. It simply responds with changes in heart rate. So organ system functions. So we looked at that one parameter, but we can also see organ system functions that are integrated as well. So for instance, when we look at the control of blood pressure, there are factors that affect blood pressure that come both from the cardiovascular system, so we can change the cardiac output, as we just said. We can change the resistance with the blood vessels. We can actually move blood from the venous side of the circulation over to the arterial side to bring pressure up if we need to. But we also have mechanisms that work through the kidneys. And so now we're starting to see this overlap of systems in the control of a single parameter. And this is just a figure showing you that how that would work. So you see that you've also got behavior that comes in. And so that would then be part of the brain. So if your blood volume and blood pressure are down, uh, you're likely to get thirsty and want to go drink water, which would help bring the volume back up. Here's another example where control of pH in the plasma, another one of our regulated variables, depends upon two systems: the respiratory system, which can ventilate CO2 off, and in doing so, cause a shift by law of mass action and get rid of hydrogen ions. Or we have the kidneys that can both reabsorb or secrete bicarbonate and H plus to change our buffers or to directly change plasma pH. So here we see another example where we have two systems working hand in hand to maintain homeostasis of a single regulated variable. And now we get to the last part, which is the integration of systems and how we can see that in pathologies. So here's our pathophysiology part. And understanding the mechanism of what is going wrong is critical for treatment, because if there is a mechanism that has gone wrong and you have a way to correct it, that is the best way to do it. So, for instance, if you have someone who is a diabetic because they don't have insulin, what's the problem? The problem is not enough insulin. You would correct it by giving insulin. You wouldn't treat the symptoms necessarily unless they are life-threatening. You treat the underlying problem. So we'll look at two examples. We'll look at dehydration, and we'll also look at diabetes. Now, I will warn you that these are very complicated figures, and you're not supposed to see them directly, to see all the details in them. So this is our dehydration map where we have, these are the circles at the top. The yellow circles say decreased blood volume, decreasing blood pressure as a result, and increase in osmolarity. And what you see in the colored vertical bars are the responses from multiple systems. So the peach colored one on the left is the cardiovascular response, which is primarily aimed at trying to bring blood pressure back up. The purple one is a response that ends up with the production of angiotensin 2, which goes everywhere to try and bring things back to homeostasis. Then we have the direct effect of decreased blood pressure on GFR at the kidneys. That's the beige colored one. And then finally, we have the hypothalamic mechanisms that include thirst as well as the production of vasopressin, which is aimed at reabsorbing water maximally in the kidneys. So all of these are trying to work together to bring osmolarity down and blood pressure up. So this is also an example of our mass balance issue. Understanding mass balance, if the pathology here is there has been loss of dilute body fluid, That's why the osmolarity went up, and the volume is down. That's the fluid loss. The appropriate treatment is replace what was lost. So this is that idea of mass balance. If you've lost something to go back into homeostasis, you replace what was lost. And in this case, that would mean a hypoosmotic fluid, drinking water, for example. So here's the second one. And again, another very complicated map that you should not be trying to read. And it's essentially a type 1 diabetic who is in ketoacidosis from lack of insulin, from lack of insulin. And what you see is you end up with hyperglycemia, and now you've gone into the dehydration part that we just looked at because of osmotic diuresis with the loss of glucose in the urine. And then you end up with the low blood volume, low blood pressure combined with a metabolic acidosis. And so, what we see when we get something like type 2 diabetics, we may not see this extreme, but we see more of the interlapping and integrated function where they are going to have changes in multiple body systems, all because of the problem that they have with insulin resistance or lack of insulin. So you see changes in the eyes, in the nervous system, in the kidneys, in the vascular system, in the intestines with slowing of the stomach. So, changes in one system then often will cascade and create a problem in another system. And this is why it is so critical as you study physiology, and particularly your pathophysiology, to remember this integrated function of the body's systems. So this is going to help you as you go forward with your studies, and I hope that you will remember some of these core concepts as well as some of the mechanisms. And remember, always physiological function is integrated. So thank you for your attention, and I'm going to come off of screen share so that I can see you, and perhaps you have some questions. So I see Muhammad raised a hand. Yes. Did you want to ask a question? Let's see. Okay. I think your volume is really low. Can you raise your volume?
Can you hear me
Or
now?
actually, let's see. Where's
Okay.
we
Uh,
have
okay,
Here we
so
go.
I'm acting as moderator for this discussion session or question and answer session. And on my screen, there is Radin Robani. Maybe you wanna ask a question and go ahead.
Yeah, please, Raden, you can open your mic and ask for the questions.
Baik, thank you for the explanation, Professor. May I ask question? How do temperature, blood pressure, and uh, pH work together to maintain homeostasis in the human body? Thank
They
you.
don't they don't work together necessarily, but the they are different parameters that will have to be maintained in homeostasis for the body to stay functioning. So the different regulated variables don't necessarily work together. But for instance, there are sometimes challenges. So if you are exercising, let's say you go out and you exercise and you sweat a lot and you have lost blood volume and blood pressure, then that is the sweating was an attempt to maintain temperature, right? That's why you're sweating, you're trying to cool your body. And at some point, the integrating center, the brain, has to decide, am I going to maintain blood pressure and still get oxygen to the brain, or am I going to maintain trying to keep the temperature down? And this is where you start to see examples like heat exhaustion and heat stroke, where the body simply can no longer maintain the temperature homeostasis because it's trying to maintain blood pressure homeostasis and blood flow to the brain. So sometimes these factors become conflicting with each other. I hope that answered it.
Thank
Yeah.
you, Prof.
Thank you, Radin, for the question. For the next question, maybe from Gamma class, Muhammad, please.
Okay, thank you. Good morning, Professor D. Silverthorn. First of all, let me introduce myself. My name is Muhammad Zarandarmala as a student from Faculty of Medicine, University of Sriwijaya. And I would like to express my deepest gratitude for the opportunity to learn and be taught with such excellent and beneficial materials. Professor, I have a question, which is how can the integrative science approach in a physiology help in understanding the interactions between various human body systems, such as the nervous system as the fat control system of human body, cardiovascular and endocrine systems. In response to external and internal environmental change, and how can this understanding be applied in the fields of medicine, sports, and public health to improve human quality of life? Thank you, Professor. That's all.
Oh, that's all, but it's a big question. I mean, it is very important that we
Yes.
be thinking about all of this as we're
Okay.
going
Thank you,
forward.
Professor.
You're going to be seeing a lot more as I don't know what climate change is doing to Indonesia, but for instance, in our country and in Texas, we are seeing much hotter temperatures than we've ever seen before in the summer times, and regularly over 40 degrees, 42 degrees, and many days. And people have to change behavior, as well as the medical community must be more aware of the problems such as heat stroke and uh, and heat exhaustion that will be coming in to see them in the emergency room. So you asked a very big question that I don't have a single answer for.
Yeah, thank you, Prof. D, for the answer. Yeah, of course, the climate change also change our behavior, like you say. In Palembang, also the hot is, I think the temperature is also higher than before, than we were as a child. So yeah, the heat stroke is real.
Yeah.
Is there
Yeah,
anything you want to add?
yes. Like
Prof?
I want to add that, like in Indonesia, in tropical countries, uh, we feel that there is no more different season. Like before that, in every December is a rainy season around December, before or after December, and then in August is a dry season. But now there is no season like that.
Really?
Today,
Oh,
yeah, we feel that this is rain, big rain, and then hot, and then big rain again, all the years. So this is very. I remember Al Gore ever 10 years ago ever said like that. He said in tropical countries there will be no more like that rainy or a dry season in every year like before, and it's already happened. Like Al Gore said 10 or 15 years ago.
mm,
This is what want to say to you.
yeah. Do you, in Indonesia, do you have an effect from El Nino and La
Yes,
Nina?
yes, yes.
Yes, we're
No,
seeing
yeah,
big effect.
yeah. It should be no more rainy season in March today. But
Mm,
now in Indonesia, that if we have a big flood in several area, especially in Jakarta and Java. So it is already crowd, not regularly situation anymore. Yeah, and the other part is very dry, but in not too far from that location is heavy rains, something like that.
Yeah.
Hmm.
We're having very dry, and as a result, we're having terrible fires. I'm sure you all heard about the big fires out in Los Angeles, but now we have them all over Texas and Oklahoma because everything is so dry. We're just not getting the rain that we're used to.
Wow. Yeah. Thank you.
Okay, we move on to the next question. Oh, uh, from the Alpha, Muhammad. Sorry, I cannot see your full name.
Muhammad Dwi Fo.
Muhammad Wifo.
Thanks for giving me a chance to ask. So I'm quite curious about the homeostasis part of your explanation, where you mentioned about the dash line or the desired range. So the question is, can we actually make the line becomes wider or increasing the tolerance of our homeostasis? And can it improve our quality of life, like the tolerance of the pathogenesis or pathology?
Yeah. That's a very good question. I think, can we intentionally change it? I don't know that we have the ability to change it intentionally, to manipulate it ourselves. But we see, for instance, in exercise, there's it's variability around what we call a set point. So you've got a line and then it's plus or minus, just like you do in chemistry, plus or minus 2 degrees or something like that. So you have a set point, and when you exercise, your set point for blood pressure homeostasis actually moves up. And we don't know exactly how that happens, but set points can change for an individual. And there are also genetic determinants of set points. And so, not so much with humans that I know of, but I certainly know in the animal kingdom, you will start to see, as the temperature is warming, that the animals that have higher set points or a wider range then are favored for reproduction. And that is probably happening to some extent in the human population. We just have no way to track it. And I don't know that anyone has ever thought about actually trying to change our set point or our tolerance, increasing our range. But it may be something that will happen over time.
Thank you, Professor.
Yeah, maybe it's the example of the athletes. How about it, Prof? We know athletes have, like for the example, the swimming athlete, maybe they have a different lung capacity, I think. Is it something changing in their body due to exercise or something like that, Prof? What do you think
Some,
about it?
yeah, some of that will be training, and some of that definitely is genetics. I mean, the people that are doing it have inherited a better lung capacity or the ability to train to a higher level. You can look at, for instance, the origin and insertion of muscles and predict that people that excel in certain sports are going to have different origins and insertions for their muscles because it will give them a biomechanical advantage. And so I think that's what we're seeing with the athletes. Yes, there's a certain range that you can train yourself for, but there will be at some point a genetic limit beyond which an individual can't train more.
Okay, Professor. That's very interesting. Thank you for the answer. Yeah, if there is more question, Nur Hafia, please.
Okay, thank you, Dr. Raisa. Good night, Professor Silverthorn. Thank you very much for giving us such a wonderful lecture to me and all of us. It has been an honor to meet you, so I'll not waste this opportunity to ask questions. So, during the lecture earlier about controlling systems, basically focus about sympathetic and parasympathetic. So, in your lecture and in class and some of the literature that I read, which is actually not that much, but giving me impressions like sympathetic and parasympathetic are two things that are always work antagonistically and at two different times. So, I'm just wondering, if it's possible for sympathetic and parasympathetic to work at the same time with the same volume, with antagonistic roles or with non-antagonistic roles. Thank you, Professor.
Oh, very good question. And I know let me think about it. I know there are some examples where sympathetic and parasympathetic both work together to accomplish the same endpoint. And I'm trying to remember what the example is. So they're not always antagonistic. I mean, for one thing, there are some systems that are controlled only by one division and not by another. And I will we'll see if it pops up in my mind. But no, there are some examples where they work together or synergistically to help achieve the same goal. And Irfan, if you can remember what the example is, please chime in. So
Yeah, it's very hard questions, I think, for
yeah.
me. So I cannot.
Yeah, no, I know there's an example, and it's something I should be more ready to answer, but very good question. Yeah.
Most of the time they work antagonistic, is it, Prof?
Or a system will have control by one and not by the other. I mean, not everything is antagonistic. If there's dual control, I will send Prof. Irfan when I find the example. I will email him and he can email it out to all of you so that you know what it
Okay.
is. How's that?
Yeah. Yeah.
Okay.
Prof Irfan, maybe you want to say something about
Tentang
this?
apa tadi? Saya belum begitu nyaman. Itu tadi tentang apa tadi?
Apakah itu, Prof, simpatetik dan parasimpatetik itu bisa bekerja bersama-sama, Prof? Kurang lebih seperti itu.
So, actually, in some complex activity, I do believe in, but I'm not sure about that. I'm not sure about it because it is sympathetic and parasympathetic is controlled by centralistic system in the autonomic system, probably. Yeah. So maybe not together in the same time, but they can be changed in shorter time. I think like accommodation, the increasing sympathetic, and then like exercise. I do believe when they do the intense activity, like in exercise, we have example interval training. Interval training is that they can do the high intense exercise with the higher sympathetic activity, and then suddenly they slow down the intensity, and the parasympathetic become more dominant. For now, I can answer now, but later
Yeah.
I will looking for another example. Yeah.
Yeah.
Thank
It was a good question, a very good question.
Yeah, we're looking for Prof the answer that we can. For now on, we have still 4 raised hand. Please, Mas Abdullah.
Okay, assalamualaikum warahmatullahi wabarakatuh. Good morning, ma'am.
Good morning.
So, I have a little question. Our regulated variable. So, is there any chance our body did not giving any action when there was outside desired range stop?
I'm not sure I understood that.
Sorry, can you repeat the questions?
Yeah.
Okay. So, is there any change outside desired range, but it goes into no action required? Thank you, ma'am. Well,
Yeah. Can you type in the chat for me the question to make sure I understand it? On my end, your volume you break up a little bit. It's
Is typing the
Bisa dong bahasa
No.
Indonesianya, Mas. Mas coba bahasa Indonesia, kita coba terjemahin.
It's okay. I'm gonna type in it, sir. Hold on. Okay, doctor, hold on.
Okay, while Kemas is typing, we move on to the next question first. Beta Saulia. Please, Saulia.
Yeah. Thank you so much, the professor, for the lectures. It's a very wonderful opportunity to be able to learn from you. It is mentioned previously that a small change in protein can change its binding properties. But I would like to ask, why exactly does that happen? And can these alterations be reversed naturally by the body? Thank you.
These are generally determined genetically, and so it's literally a change in the amino acid sequence, which will potentially change the three-dimensional structure of the protein, or it could change the binding affinity at the binding site. For instance, sickle cell anemia is an example where the only one amino acid in this very long chain of amino acids has been altered, and yet it changes the shape enough that it causes the disease. So it's genetically determined, and of course now there are many people working to try and see how we can reverse these genetic changes by using CRISPR or something similar to actually put in the corrected code for the protein. But it can be as little as a one amino acid change that changes either the shape of the protein or the binding affinity or specificity.
Thank you, Prof. D, for the questions.
Okay, so I think we have, is there any chance, this is from Kimas, if I've said it, Abdullah, is there any chance for an outside desired range to not being detected so that nothing happens? Well, that would be potentially a pathology. So yes, if the sensor, if something has happened to the sensor, for whatever reason, the sensors are cells and groups of cells, so you could have something like loss of blood flow and oxygen that kills the sensor, in which case, yes, you would have the monitored variable could go outside the range and not not sense the change and not cause a response. But that would fall in the category of pathology, not physiology. Thank you for typing it. I see there's another one. Let's see, is that right? Yeah. No, that's it. Okay.
Okay, next question from Kaif Gustav, please.
Thank you for the opportunity. It's just a quick question. I want to ask, what's the risk of complication for people with hypothermia when they get sudden change of body temperature from low to high? Thank you.
Did you say hyperlipidemia?
Hypothermia.
Oh, hyperthermia, hypothermia,
Hypothermia. Hypothermia.
hyper.
Yes.
Okay, so
Hypo.
Temperature, Professor.
yeah,
Hypothermia.
Lower
hypothermia.
hypo,
Okay.
hypothermia.
Okay. So, and then what happens if the temperature? I'm sorry, I've now missed the question. So sorry. If you
Uh,
could, uh,
Please, can you make the questions maybe a little bit shorter and
Uh,
ask?
so
what's
if
the
a
risk of
person has hypothermia, is that what you're saying? Someone has hypothermia and
the risk of complication when they get sudden change of body temperature from low to high?
good question. I'm not, uh, how would that happen? I mean, one of the things that if you have someone with hypothermia, one of the things you do is you warm them very gradually because it could, I mean, cells don't like a shock. Um, and you can actually totally, particularly if the cells have become adapted to something lower, you could potentially, if the temperature goes too high, you could have, say, a protein structure. If it was a long-term hypothermia, maybe someone just has a normal low body temperature, and then they suddenly get a fever. This is why high fever is bad, because it will change the shape of proteins. It can denature proteins and hurt them. That would be the only thing that I could think of in terms of the rapid increase is that the cells have become used to low temperature, and now uh, it's going to hurt something in the cell that is adapted to the low temperature.
Okay, thank you so much, Professor.
Thank you, Prof. D. Another questions from Indah.
All right, thank you so much. So I would like to ask about the concept of losing weight and homeostasis. So when it comes to losing weight, caloric restriction is the key. So can caloric restriction permanently change metabolic homeostasis, or are they just temporary? Thank you.
Well, one of the things that happens is calorie restriction, yes, can change metabolism. Um, and at the same time, if you're restricting calories because you're overweight, cells seem to develop a set point where they want to have the adipose tissue has reprogrammed to a new set point, if you will, where it wants to stay at that set point. And sometimes if you restrict calories, you lose weight. And then when you stop restricting, you go right back to the previous weight because something in the body has reset to, and I'm going to say it's been programmed somehow to try and return the body to that set point. And did that answer the question?
Yes,
Yeah.
thank you very much, Professor.
So actually losing weight is much more work than only caloric restricting, is it, Prof?
Yes. Yes, and it changes with time. I mean, for women, changes in hormones will change the metabolism and the set point. So it's very difficult in many ways. all of these people are losing weight now with the GLP-1 drugs, and then they're discovering when they go off of them, they go right back to where they were before.
Okay, we move on to the next question. Radin Rabani.
Thank you for giving me a chance to ask questions before, Prof. What a great opportunity to ask the professor directly. I want to try to answer the previous question. Can the sympathetic and parasympathetic work together? I think these two systems work not at the same time, but the systems work in turn and in balance to ensure that the body can respond to a different physiological needs or homeostasis maintaining balance. For example, after the body responds to stressful situation by increasing heart rate and blood pressure through the sympathetic system.
You're breaking up and I can't understand anything you're saying. I'm sorry.
Yeah, Radin, maybe you can type the questions because your
yeah,
yeah. Please type the questions and we move on first with Zilan Sarif.
Okay, first of all, I would like to say thank you to Professor Silverthorn for giving us the material for today. I would like to know more about the extracellular vesicles communication. I remember you said that this is for us to explore more, so why is it gaining more attraction now, and why do you think it's beneficial for the future? Thank you.
Well, it's something that we've never known before. It turns out there are many different types of extracellular vesicles that are put out, and we're still discovering what a lot of it is. In some cases, if you know the term budding, budding, the outside of the cell comes up and makes a part that breaks off and goes. So that's one way. So the inside of the cell has now turned into a vesicle that goes through the bloodstream. And then in another form, it actually occurs inside the cell in a big vesicle inside the cell that has many little vesicles in it. And that goes over to the cell membrane and opens up and lets all these little vesicles go out. There's a lot we know less about it, what it's doing than we know about how it's doing. And so we're just now discovering, but this is how one set of cells in the body apparently can communicate with another tissue or organ. And such a new area. It's not my specialty, but it's very, very interesting and something to keep your eye out for.
Okay, thank you, Professor.
Thank you, Professor. We still have so many questions, Professor.
That's okay.
Okay, next one is Ihsan, please.
Okay, thank you very much for the opportunity, Prof. Here's my question. Faced with sudden changes in environment, such as facing unexpected storm in high altitude or a sudden drop in temperature, what is the core and most important mechanism or part of the body that can help in maintaining the homeostasis in case if it is possible at all, and how does it work? Also, in does kind of scenario, is there a way to prepare in advance so that we can face changes that usually will cause problems if being faced head on or something like that?
So let me the general answer is many of the changes go through the brain. I mean, we have many different centers in the brain that monitor some of the parameters you were talking about. And in terms of can we prepare for them? A good example would be high altitude, because for each of the situations you mentioned, it's a different factor that is changing. And so going to altitude may have two changes. It may be lower temperature, and it may also be low oxygen. And the response then is going to be to try and increase the ability of the body to transport oxygen by making more red blood cells. And so can we change that? Yes, we can, but it requires not going from sea level to the top of Mount Everest. It would be gradual changes to allow the body to sense that oxygen is going down and that more red blood cells are being made. So in some cases, it is possible. We call that acclimatization is the term. If it's done out in the environment where you expose yourself, if possible, to slowly changing environmental situations. Another example is people who go and work outside in hot environments. Over time, they will start to make, initially, their sweat will have a lot of sodium in it. And then with acclimatization, they will be putting out more aldosterone and actually reabsorb more sodium from their sweat. So their sweat becomes more and more hypotonic. So acclimatization can happen, but again, only within the genetic limits for that individual. So let's see. I see we got our question in the chat. Oh, wanted to try and answer the previous question. Can they work together? Can work in turn and in balance? Yes, it just depends. Do you have a chapter in your book on the autonomic nervous system that shows, often it's a body and it'll show sympathetic going to this side and parasympathetic going to that side? That would be a good figure to look at and see what organs are affected by only one system and what are affected by both. But yes, it could be very well be that in this situation, sympathetic is working, and in a different set, that same organ would be under the control of parasympathetic. Yes, so it is possible that they work alternately as well as immediately antagonistically. And your example of heart rate is a good one. Yes.
Thank you, Radin.
Thank you
Thank
for
you, Prof.
typing it.
Yeah.
And thank you for typing it. It's much easier.
It's a long time. Okay, Zofir, sorry, please.
Okay, thank you for the opportunity, Prof. My name is Zofir Gisaldi Gusan. I have a question. The question like we already knows that the intracellular fluid has a greater amount than the extracellular fluid. So my question is, how does the greater volume of the extracellular fluid compare to the how does the greater volume of intracellular fluid compared to the extracellular fluid contribute to maintaining homeostasis in the human body? Thank you.
So, yes, there is a total greater volume, intracellular fluid volume, but it is what is monitored for homeostasis is that extracellular fluid volume, because it is every cell will be exposed to the extracellular fluid. And so it doesn't matter that the volumes are that different. It's actually much easier for the body to try and maintain homeostasis in a smaller volume. Plus, each cell, the cells are not, we talk about intracellular volume being bigger, But what we've got are billions and billions of tiny little cells with very small volumes, and they are each independently, for the most part, communicating with the extracellular fluid. So it's much easier to maintain homeostasis of the extracellular fluid.
Thank you, Prof.
Thank you, Prof. D. I'm interested in what you said before about the extracellular vesicle. That's new things for me to hear.
It's a new thing, a very new thing for me too. I've been looking at it. I've been working on the next edition of my book, so I had to read up on it. But it we talk about it, but I haven't really put examples in because it's not. It's still what we would call an emerging concept. So, for instance, in multiple editions of my book, many years ago, I had a book. We have boxes called emerging concept, meaning these are new hot topics, if you will. And in an early edition, it was stem cells. Stem cells were in this little box. And then the next edition, 3 or 4 years later, they got a little paragraph in the text itself. And now, of course, they're a whole section at the beginning, and they come up in many chapters. So uh, extracellular vesicles are like that. We're just at the beginning of learning about them, and it's something that you, as up and coming physicians, can pay attention to and see because it may be an exciting area for new therapies.
Yeah, it's good to know that the physiology field is also a growing field. It's not only about the pathophysiology, but the physiology is getting more and more exciting. And I think we can um have three more questions, the last three questions, because it's already 9:30 p.m. in you.
That's fine. Yes, 3 more questions would be great.
Yeah, okay. The last three is, please, Cindy.
Okay, thank you, Doctor. Good night, Professor. My name is Cindy Febra Damianti, and I'm from class Alpha. So thank you for your explanation about homeostasis. I have an experience that sometimes bothersome based on my experience and what happened in my body, and I think it might be due to a failure in homeostasis compensations. So may I ask Professor about it? So could you kindly explain how cold and hot external temperatures might lead to inflammations or the release of histamine in the skin, giving me the itchy skin? So thank you, Professor.
Yeah. I am not a physician, so I'm just telling you from my experience, but essentially, some people develop what is an allergy to temperature, and to hot or to cold, and it can be very extreme in that sense. But something has happened with the immune cells that they have become sensitized to the temperature. And when you experience it, then they start to release histamine, the mast cells. And it's probably this communication between the thermoreceptors and your immune cells that is triggering it. But it's not uncommon for that to happen, that people can develop an allergy to temperature.
Yeah, you can learn more about the hypersensitivity type maybe later. Yeah. Um, the next questions is Izati.
Okay, thank you, Dr. Resha, for the opportunity, and good morning, Dr. Day.
So my name is Izati Laksmana and I want to ask about erythropoiesis. So we know that iron is the one of the main source for blood production in bone marrow. So I want to ask when the Fe3 plus go through intestinal cell in duodenum to get converted to 2 plus state and then got converted again to 3 plus state and then it will send to bone marrow. So why the if Fe3 plus must go through intestinal cell first when the end of the process is still the same state?
You broke up a little bit. You're asking about bone marrow and blood cell production, but I lost the middle part.
So we know that iron is the one of the main resource for blood production, and we get iron from our digestive system. So as I remember, it called Fe3 plus. So it's on the 3 plus state, and it must go through the intestinal cell first in duodenum. But the end of the process after ejected from the intestinal cell, it remained the same state. So why it must be go through the intestinal cell first? Why don't you just go to circulate it on the blood? Thank you, sir.
Well, it has to, I mean, to get into the body, it must cross the intestinal cell. And it turns out iron cannot pass through a phospholipid bilayer, so it actually is going on transporters. And there are a couple of different transporters. So you must have a transporter on the apical side that faces the lumen. And then you also must have a transporter on the basolateral side. And then there are several hormones involved in this. And there's a transporter for the blood that is different. So it's quite a complex mechanism, iron homeostasis and metabolism. So it's not simple. It can't just, and remember that when we have to maintain electrical neutrality. So whenever you're absorbing the iron, you must also be absorbing an anion that comes along with it.
Okay, thank you, sir, for your explanation.
Thank you. The last question, Insira, please.
Okay, thank you for. Okay, first of all, I want to ask, what are the people who are able to withstand stress and be able to adapt? Kan kayak homeostasis tuh, I mean homeostasis can adapt. But why some people cannot? Like for example, surviving disease such as COVID, but while others fail to survive and suffer fatal repercussion and even die. Thank you.
I think it's simply a matter of genetics. We all inherit a very different set of genes, and some people inherit the genes that allow them to adapt, and other people have a more narrow range of homeostasis, and they simply cannot adapt. But I think it's partially the acclimatization, if they've been able to acclimatize to something, and then also then it's the issue of the genetics. Do they have the genetic flexibility to be able to adapt? And I think that's I would guess that genetics play a very large role in that. And possibly the other thing would also be if they have underlying conditions. I mean, that's, what their health. Is their health good in general, or do they have diabetes or some other condition that makes them less tolerant and less able to adapt? But if they're all healthy, I would say it's probably genetics.
Yeah. In during COVID situation, I think the people who got fatal condition is the one with underlying diseases or underlying immune diseases.
Mm-hmm.
Yeah.
Yeah, yeah.
Prof. Irfan
and
tadi?
also about the past experience that they got, which mean, for example, for another example, like say that in hypoxia condition, if the person have already trained to practicing in hypoxia condition, so he will be able to adapt in that stress situation, while other cannot because he have no experience to train about it. I think this is my additional answer. Thank you.
Yeah, we already came to the end of the class. Miss Prof.
Yeah.
Prof. Irfan, is there any?
Maybe you can ask with the text, and then Professor D will answer it by email. I think we can send email to Professor D because it is already late night in America. So maybe Professor D want to take rest. Yeah. Yeah.
So yes, it has been a pleasure. I love the questions. And yes, if they would like to I think email them to you, one of you, and you can send them on to me and I will try and answer. And I will find my example where sympathetic and parasympathetic work together. I know there is one because this question has
Okay.
come up. But I will send it to you to send to the class then.
Yeah, thank you
Then.
very much for that, Prof. D. Bisa tutup, Prof? Atau?
Ya, silakan, nanti terlalu malam.
Yeah, because it's already late night in Texas. Um,
And
I
later in New York, so it's an hour later.
I want to ask a little more things about your latest work about Physio K Fun. I found it in your Google Scholar.
Yes, it's a set of curriculum guidelines that we developed for the African universities because many of them don't have strong physiology departments or programs. And so it's a set of suggested learning outcomes where no one can possibly teach them all, but you decide what your program is, and then you pick the ones that are appropriate for your program. So,
Yeah.
do you find them useful, do you think? Did you?
I haven't read the full text about it. Maybe I will try it.
Yeah.
It can beneficial to our university also. Yeah, thank you very much, Prof. D. Silverthorn, for the amazing time. Thank you for your time. I close this presentation. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi
Maybe one last message from Professor D to our student. Professor D, please give a last message how to be success in learning physiology, I do believe. Please, Professor D.
I think you should. One of the things that's important for physiology, but also very important for medicine, is this ability to recognize patterns. And they have studied how physicians make a diagnosis. And what they realized is that they are doing very rapid subconscious pattern recognition, that they have seen so much material that their brain is working fast, their unconscious brain is working faster than their conscious brain to look at the patterns. So learning what the patterns are will make your life so much easier because when you find something that meets the pattern, you already know it. You just have to think, oh, well, this one's a little different. And all you have to remember is that one little difference because it matches the pattern. So having the patterns in your brain make it much simpler to go, oh, well, that fits that pattern now. It's just part of this list that I put under that pattern. So learning the core concepts and recognizing them, I think, can make your life much easier.
Okay, thank you. Oke, Raisa bisa ditutup sekarang.
Yeah. Thank you very much, Prof. D. Good night.
It was my pleasure. Good night to you all. And I hope
Okay,
that
thank
perhaps I
you.
will have a chance to return to Indonesia and meet
Yeah,
some of you.
hope so.
Yeah,
And
forward to
Yep.
meet you
So
in
bye
person.
bye,
all right,
Professor D. Thank you
yes,
so much,
yes.
Professor D. See
All
you next
right.
in Germany, Frankfurt.
And Germany, yes, right. So are you
Okay,
coming to
September.
Germany?
Okay. Yes, I
Yeah.
will come.
Okay,
Raisa,
good.
I hope you can go so also, Raisa.
I have a
Yeah.
babysitter.
Okay.
Okay,
Bye-bye.
bye
Bye.
bye. Bye bye. Thank you, Professor Dit. Terima
Thank
kasih
you,
adik-adik ya.
Professor.
Pertanyaannya banyak bagus-bagus ya.
Thank you, Prof.
Yuk kita tutup.
Uh,
Yuk, selamat malam, selamat pagi semua. Assalamualaikum.
Salam.
Makasih, Dok.
You're welcome.
Untuk yang di rumah cukup ya.